Now, let's discuss meiosis in more detail. Specification number 9 asks us to describe meiosis as a reduction division in which the chromosome number is halved from diploid to haploid, resulting in genetically different cells. At this level, the detailed stages of meiosis are not required. Meiosis is a type of cell division in which a parent cell first replicates its DNA and then divides twice to produce 4 daughter cells. Unlike mitosis, these daughter cells are genetically different from each other and from the parent cell. During this process, the chromosome number is reduced from diploid, which has 2 sets of chromosomes, to haploid, which has only 1 set. This reduction is essential for producing gametes with half the chromosome number, so that fertilization restores the full diploid number in the zygote. We define meiosis as, meiosis is a reduction division in which the chromosome number is halved from diploid to haploid, producing cells that are genetically different from each other and from the parent cell. In simpler terms, meiosis reduces the chromosome number by half to form haploid cells, ensuring genetic variation in the gametes while maintaining the correct chromosome number in the next generation after fertilization. Meiosis is a type of nuclear division that produces genetically unique cells and is responsible for the formation of gametes, or sex cells. During meiosis, the chromosome number is halved, so the resulting gametes are haploid. This reduction is crucial because, without it, fertilization would create a zygote with twice the normal number of chromosomes. For this reason, meiosis is called a reduction division. It transforms diploid cells into haploid cells while also introducing genetic variation, ensuring that each gamete is genetically different from the others. In the process, the parent cell first replicates its DNA, then divides twice to produce 4 daughter cells, each with half the chromosome number of the original cell. The structure of chromosomes in meiosis begins with each chromosome replicating before the process starts. This replication produces 2 identical sister chromatids joined at a centromere, forming the characteristic X-shaped structure. These replicated chromosomes then pair with their homologous partners, one inherited from the mother and one from the father. During this pairing, sections of DNA can be exchanged between the maternal and paternal chromosomes, creating recombinant chromosomes and introducing genetic variation. Meiosis is unique because it involves 2 consecutive divisions. In these divisions, only 1 copy of each chromosome is passed to each gamete. As a result, 4 haploid daughter cells are produced, each containing half the chromosome number of the original parent cell. The way chromosomes pair, recombine, and separate in meiosis is distinct from their organization during mitosis. Now, let's discuss the process of meiosis. Meiosis begins with a single round of DNA replication, followed by 2 consecutive cell divisions. During the first division, homologous chromosomes pair up along the center of the cell. While paired, sections of DNA are exchanged between maternal and paternal chromosomes, creating recombinant chromosomes and increasing genetic variation. The spindle fibers then pull the homologous chromosomes apart, so each new cell receives 1 chromosome from each pair. In the second division, the chromosomes line up again along the center of each cell, and the sister chromatids are separated, similar to what happens in mitosis. At the end of meiosis, 4 haploid daughter cells are produced, each containing half the original number of chromosomes. This process differs from mitosis because of the pairing of homologous chromosomes and the recombination that occurs during the first division. Now, let's watch the video to get a clearer understanding of how meiosis works. It starts with the cell copying all its DNA so each chromosome has 2 identical strands. In the first division, similar chromosomes pair up, and segments of DNA are exchanged between them, creating genetic variation. The spindle fibers then pull these paired chromosomes apart, and the cell splits into 2, each with half the original chromosome number. In the second division, the chromosomes line up again, and this time the identical strands of each chromosome are separated. After the final division, we end up with 4 unique haploid cells, each containing half as many chromosomes as the original cell. This process differs from regular cell division because of the chromosome pairing and DNA recombination that happens in the first round. So, finally, the importance of meiosis can be understood as: meiosis is responsible for producing gametes, including sperm cells, egg cells, ova, and pollen grains. During this process, the chromosome number is reduced by half, so the gametes are haploid. This reduction is essential because fertilization restores the full diploid chromosome number in the zygote. Meiosis also generates new combinations of maternal and paternal chromosomes through recombination. Combined with random fertilization, this ensures that each offspring is genetically unique, increasing genetic variation within a population.